Nearly 10 million people are parasitized by the tapeworm Diphyllobothrium Latum in the cooler regions of the northern hemisphere. Those who harbor the parasite may develop a blood condition resembling pernicious anemia. This tapeworm requires 3 hosts to complete its life cycle: a fish-eating mammal such as man or the dog, and 2 aquatic animals, a copepod, one of the crustaceans, and a fish, usually a species of pike. The adult worm is located in the small intestine of the mammalian host. In the copepod, a small larval stage develops, while in the fish host is found the larger and final larval stage. The adult Diphyllobothrium latum is ivory-colored, attains a length of from 3 to 30 feet in the intestine of man, and may live there for a year or more. This parasite is called the fish tapeworm or the broad tapeworm because of the width of its segments or proglottids. In the intestine of the host, the adult tapeworm may release hundreds of thousands of eggs daily. When infected people defecate in or near bodies of fresh water, or when raw sewage is discharged into these waters, the eggs become a potential health hazard. The egg averages about 70 microns in length and 50 microns in width. At one end of the light brown shell is located a lid, the operculum. And at the opposite end there is generally seen a knob-like thickening of the shell. The egg is filled with a mass of yolk cells. As development occurs in fresh water, the early embryo stage is visible. Continuing growth, the embryo becomes spherical, almost filling the egg. The embryo begins to show movement, especially of the hooks, which are characteristic of tapeworm embryos. After a week or more of development, the embryo is mature and it hatches. Left behind, yolk debris and the vitelline membrane are forced out of the shell. The operculum remains attached. The hatched embryo is known as a coracidium. Newly hatched Coracidia swim rapidly and have a rather elongated shape. Later, they become spherical and their movement slows down. Coracidia may spin or become stationary. Here the hooks may be seen again. There are 6 hooks with sickle-shaped ends. The movement of the coracidium in water is caused by the beating of cilia present on the surface of the spherical larva. The action of the cilia may be studied to better advantage in slow motion. The cilia are much longer on one side of the larva. When the coracidium is moving forward, the long cilia are at its anterior pole. Each coracidium has two small flame cells with vibrating cilia. These structures are part of the primitive excretory system. Coracidia consist of two parts, an outer ciliated envelope, the embryophore, and the inner true embryo, or oncosphere, containing the hooks and flame cells. The coracidium is the only free-living motile stage in the life cycle of Diphyllobothrium Latum. Actively moving, these organisms may live for several days in cool water, even though they lack a digestive tract. The first aquatic host of Diphyllobothrium Latum is a copepod. Several species of the genus Diaptomus and a few species of other genera are susceptible to infection. Coracidia are ingested by the copepod. In its stomach, the embryophore, or outer ciliated envelope, is ruptured, releasing the oncosphere. The six-hooked oncosphere becomes active. Pushing against the intestinal wall with the hooks, it breaks through into the blood sinuses of the copepod. Here, the oncospheres round up and become almost motionless. These larvae are now called procercoids. They begin to grow and are considerably larger in about 5 days. After 2 or 3 weeks, the procercoids are almost half a millimeter long. They are now motile and have a caudal appendage. This caudal appendage is the cercomere. In older procercoids, the cercomere becomes more rounded. Within it are the 6 hooks seen in the oncosphere. The body of the procercoid is muscular, contains numerous calcareous corpuscles, and the anterior end is characterized by an invagination and the presence of glands. A copepod can harbor several fully grown procercoids. Copepods are an important constituent of the plankton life in streams and lakes. They serve as food for small fishes. Copepods infected with procercoids of the broad tapeworm may be eaten by minnows and the larvae released in the intestine of the fish. Or minnows containing live procercoids or infected copepods recently ingested may be eaten by small game fish. Minnows and small game fish are in turn eaten by species of pike. In pike, the common fish hosts of the broad tapeworm, the final larval stage develops. This larva, the plerocercoid, lies free among the muscle fibers. When removed into saline solution, some of the plerocercoids become very active. They vary in size, some reaching a length of 5 centimeters. The plerocercoid is cylindrical and glistening white in appearance. The most striking morphological feature is the partially developed anterior region bearing a dorsal and ventral groove. The lips of the ventral groove are seen. Live plerocercoids of Diphyllobothrium Latum are unwittingly ingested by man when infected fish are eaten raw or when infected fish are improperly cooked. When living plerocercoids are eaten, they survive passage through the stomach. In the small intestine, they attach by their anterior ends to the mucosa and grow rapidly. In 2 or 3 weeks, the worm is several feet long and sexually mature. Since Diphyllobothrium latum sometimes attains a length of 30 feet, the worm loops back and forth on itself in the intestine. The anterior end is attenuated and the remainder of the body ribbon-like. The whole organism is known as a strobila. The strobila is made up of 3 distinct regions: the scolex or head, the short neck, and the long body. The scolex and neck comprise only the first few millimeters of the strobila. The scolex is muscular. It has 2 grooves with membranous edges dorsally and ventrally located. From these grooves, called bothria, is derived the generic name Diphyllobothrium. The bothria are for attachment and anchor the adult worm to the mucosal surface of the intestine. Just behind the scolex is the short, unsegmented neck region. Behind the scolex and neck, segmentation begins. This is the region of the immature and undifferentiated broad proglottids. Superficially, these proglottids reveal no internal structures. Following the region of the immature proglottids, the strobila is sexually mature. The mature proglottids, which contain both male and female reproductive organs, show distinct centrally located uteri resembling rosettes. These characteristic rosettes are of diagnostic importance, Diphyllobothrium latum being the only tapeworm of man with this feature. These uterine rosettes are filled with thousands of eggs. Through birth pores, the eggs escape into the lumen of the host's intestine, as many as 1 million per day from a single worm. Broad tapeworm infections are diagnosed by finding eggs in the feces. At the posterior end of the strobila is the region of senile or spent proglottids, from which most of the eggs have already been discharged.